development is notoriously complex and costly, taking up to 10 years and more than $3 billion to bring a novel molecule from the lab bench to the market. The conventional emphasis on testing medicines in animals before evaluating them in people is a major source of inefficiency. Animal models typically fail to correctly represent human physiology, which means that medicines that appear to be safe and useful in animals are frequently toxic or useless in humans. Because of this biological mismatch, many worthless or hazardous medicines proceed through to clinical trials at significant expense, while potentially beneficial molecules never reach the market. To expedite the discovery of novel medicines and customized treatment, a better approach to simulate human biology and illness in vitro is required.
In 2010, Donald Ingber and his associates at the Harvard's Wyss Institute had modified computer microchip manufacturing technologies to develop organs on chips, which are microfluidic culture devices that mimic the complex architecture and physiological environment of live human organs. These biomimetic systems are made of a transparent flexible polymer the size of a USB memory stick, with hollow microfluidic channels coated with living human organ cells and human blood vascular cells that regulate key parameters, including concentration gradients, shear force, cell patterning, tissue boundaries, and tissue-organ interactions. Physical chemical cues are necessary for some tissues to replicate the physiological microenvironment, which promotes microtissue development and function. An integrated sensing output component or a transparent chip-based visual function evaluation system delivers potential in detecting and assembling data. A realistic organ-on-chip cell model cannot be defined or accessible without the use of microsensors to assess the metabolic status at key locations in the system. Without using people or animals, these live three-dimensional cross-sections of human organs can provide a glimpse into their internal dynamics and the effects that pharmaceuticals can have on them.
Organ-on-chip has had rapid growth since it was first theorized. Wyss Institute launched a company called Emulate to commercialize these chips and bring them into research facilities. The chips have been used to successfully mimic organs such as the lung, intestine, kidney, and bone marrow, which contributed to developing brand new drugs for human diseases. The diseases that have been studied include influenza, malnutrition, radiation exposure, and cystic fibrosis. More recently, they were used by the FDA to evaluate the safety of COVID vaccines and even in space to assess the effects of space travel on neuronal function. As these chips evolve, they are increasing in degree of diverse use across multiple disciplines, providing promising results for the future.
Humans are unique, even in their way of processing drugs. This makes human trials inefficient and inaccurate for pharmaceuticals to be widely available. Humans may be subjected to mild and severe side effects, even death. In 2005, a study showed inefficient data resulted in failure rates of 57 to 88% in phase 2 clinical trials. Organ on a chip helps decrease these failure rates by screening substances, toxicity, etc. in organ on a chip before clinical trials, decreasing the number of animal testing. Organ on a chip expedites human testing, setting the gold standard and revolutionizing the pharmaceutical regulatory system. Pharmacokinetics modeling shows the absorption, distribution, metabolism, and excretion of substances. This method allows for calculating drug dosage and concentration, and it can improve the efficacy of drugs in animals and humans. Organ on a chip's main goal is to assess the safety of drugs prior to clinical trials. 48% of the human adverse effects are predicted prior. This low number is due to the inaccuracies of animal testing that provides a model for humans to filter harmful chemicals. Studies have shown that organ on a chip provides a huge safety advantage in the regulatory process by assessing the sensitivity and specificity in drugs in order to safely advance to the next stage of human trials.
With recent developments of organs on a chip consisting of liver, lung, kidney, heart, and intestine, the new ultimate goal is the integration of multiple organs, otherwise known as human on a chip. Human on a chip is much more complex as it is classified as a multi-organ chip using microfluidic technology that will help to test how several organ systems will uphold against drug therapies and testing. The multi-organ chip is vital as the single organ chip fails to reflect the full complexities of an organ system and function. The human on a chip notion, although making progress in research, is still a far-off theory. Sourcing and obtaining biological tissue is a very important parameter of creating organ on a chip. The most common type of stem cells used are embryonic stem cells, adult stem cells, and pluripotent stem cells. Although adult stem cells can be obtained through non-invasive measures, embryonic stem cells create a more ethical issue as they must be derived from human embryos. Furthermore, to maintain a steady supply of samples to create the chips, biobanks will have to be created to store human tissue. This raises various issues of donor consent and ethical considerations that may be restrictive to research.
A 2017 study discovered that the newfound technology was less costly in terms of testing, with an approximate difference of $700,000 in comparison with conventional testing. The cost of the actual manufacturing of the chips lies in the materials used. Utilizing substrates such as silicon, glass, or plastic are what are used to create the master molds. However, it presents disadvantages as a result of reduced transparency and increased cost. By contrast, glass and plastic are optically accessible and relatively cheaper than silicon, and are increasingly being used in the manufacture of microfluidic devices. As of recent, the chip has been used to study various diseases and are currently being evolved into becoming more multifaceted for future use. Past studies saw a low success rate in relation to treatments as a result of animal testing due to various errors and inaccuracies. Whereas with organ-on-a-chip, scientists are able to conduct tests on an accurate replication of human organs. With further research and future studies, the concept has major potential in medical research and diagnosis.